All right. Hi, everyone. Thank you so much for being here. I'm very excited about today's talk, today's gene delivery seminar series for the month of September. Today's speaker, Dr. Ben Campbell, brings a remarkable blend of expertise in protein engineering. Dr. Campbell began his scientific journey with a BS in health sciences from UT Santa Cruz before joining the Salk Institute, where he used patient-derived stem cells to study Parkinson's disease. During his PhD at Weill Cornell Medicine with biochemist Greg Petsko, he studied protein engineering and developed oxidative stress biosensors and fluorescent protein reporters, including mGreenLantern and HyperfolderYFP, tools now widely used in research. After earning his doctorate, Dr. Campbell served as a senior scientist at Mira GTX in New York, where he advanced AAV gene therapy vectors for ALS, work that is now progressing toward clinical trials. Currently, as a postdoctoral scholar in Lauren Luger's lab, Dr. Campbell is engineering a palette of fluorescent protein biosensors that report oxidative stress in real time with high contrast, quantifiable fluorescence changes. His collaborative work spans across disciplines and explores the role of redox signaling in neurodegeneration, psychiatric disorders, psychoactive drug function, and neuroimmune signal transduction. We are very excited to have you as our speaker for today. And yeah, you can get started when you're ready.
Great. Thank you so much for the introduction. Yeah, very happy to be here and talk about choosing the right fluorescent protein for the job. So really the purpose of this talk is to understand that, well, we all know that fluorescent proteins are ubiquitous across the life sciences now. We could hardly do our work without them. So I just kind of want to talk briefly about, very briefly touch on where they came from, what all these choices are, and kind of how we can best use them for our applications. So first, I just want to remind everybody that fluorescence emission requires excitation light. So that contrasts fluorescence with bioluminescence. Bioluminescence requires the input of a substrate, which is then broken down, and light is emitted as a byproduct. Whereas with fluorescence, so we know we need to shine, for example, blue light on GFP, and then we get green light out. And then as soon as the excitation light stops, the emission light stops. So that's different than traditional chemiluminescence, different than phosphorescence when there's a delayed return to the ground state. And so fluorescence needs input light. So just kind of not going to spend too much time on the background of where GFP came from, although it's pretty exciting and it's worth knowing. But really in 1992, GFP was cloned and sequenced by Doug Prasher, and in 1994, Martin Chalfie used wild-type GFP in mammalian cells, simply a barely codon-optimized version, and it worked reasonably well. That pretty much changed the world and led to the widespread adoption of fluorescent proteins, including eGFP. The same eGFP that we all know and love was produced by Roger Chen's lab here at UCSD in 1996. And it only took 2 mutations from wild-type GFP to produce it, which is why that one became the first and the most ubiquitous. Very quickly afterwards, there was a crystal structure of GFP. People started working on biosensors. And then in the late 90s, numerous other fluorescent proteins were discovered in coral, leading to the first monomeric red fluorescent protein, MRFP1, in 2002. And then Nathan Shaner in Roger Chen's lab at UCSD developed mCherry, and so much more happened from there. But with eGFP and mCherry, it really became possible to do 2-color imaging with fluorescent proteins. You could use blue fluorescent proteins previously, but this opened up the entire domain of red. And red fluorescent proteins have some pros and cons. It's actually been very difficult to develop red fluorescent proteins compared to the green ones. And that's a big part of this talk today, is to understand what the trade-offs are with different RFPs and what we should really use today in 2025. So just kind of going big picture here for a second, fluorescent proteins, almost all of them have the same traditional beta barrel shape shown here. So it's kind of, you can think of a fluorescent protein essentially like a can shape. In the center of the can, or think of it like a lantern, is something called the chromophore. And the chromophore is a condensation of 3 amino acids that just forms autocatalytically in the presence of oxygen. And that is actually what emits light. The purpose of the barrel itself is to protect the chromophore from bulk solvent, like the solution that it's in, because amino acid side chains project out and they support the chromophore and help translate the excitation light into emitted light. So this is why it's actually very important that the barrel be well structured and well folded, and I'll get into that a bit more later. So I like to think of the chromophore as a candle in a lantern. If you take the lantern away, the chromophore will not fluoresce. So very important to have a good, robust barrel that's properly folded. We'll get into that more. So in terms of engineering and in terms of thinking about antibody compatibility and other topics that are relevant to your research, I take great interest in this as an engineer, but there are practical outcomes of the engineering that impact your research. So although fluorescent protein structure is highly conserved, the primary amino acid sequences that make up fluorescent proteins can be up to 80% different. And that's because fluorescent proteins come from very diverse organisms like jellyfish, coral, and even anemones, and sometimes other types of fish. So they all have this 11-stranded barrel, and the chromophore tends to be in a similar conformation, but that can actually vary quite a bit. So different engineering strategies are needed, and FPs can be quite diverse, despite looking very similar. So what determines the emission of the chromophore, and this is really important for thinking about the folding and the maturation efficiency of the chromophore, which can impact your FP choices, is really just like in many small molecules, the degree of conjugation determines the emission wavelength. So in GFP, we have various degrees of conjugation between the condensed amino acids here. The DS red light chromophore has the acylamine bond, so more double bonds. And then we have some intermediate emitters like the orange fluorescent proteins that have fewer double bonds. And really what that does is it just makes the pi cloud a little bit smaller, and changes the energy gap between the ground and excited state, and we get different emissions. So the way this chromophore forms, it gets more complex as you get redder, and that's very important for our FP choices that we'll get into. So really the big breakthrough in 2004 was Nathan Shaner developing mCherry and the whole so-called mFruit series in Roger Chen's lab. And really that painted a very beautiful picture of fluorescent proteins in 2004. And this image is from Roger Chen's Nobel Prize lecture in 2008, which really showed the state of fluorescent proteins at that time. So it kind of looked like we have everything we wanted in 2008, but really what we had were version 1.0 of every color class. And this was a huge advantage. It transformed research at the time, but many of these fluorescent proteins had problems being the first of their type. And now 20 years later, we have far improved versions. And that's really what I want to talk about today. So most of the fluorescent proteins from 20 years ago can now be considered obsolete. There are superior versions that outperform them in almost every domain. So really what we're left with today is among those original fluorescent proteins, EGFP, M-orange, TD tomato, and M-cherry. These are great FPs. They've stood the test of time and they've been used in thousands of studies. So I still recommend those, but we're going to get over to talking about some newer ones. So this constitutes really a look at the visible spectrum. There are fluorescent proteins that kind of exist on the edge of that now. One type that I want to mention that are not related to the beta barrel fluorescent proteins are these proteins that emit far red that are more related to cyanobacteriochromes. They require an extrinsic cofactor to serve as the chromophore. So really, in this case, biliverdin, which is present in blood and brain, ends up being incorporated into the protein, nests into the structure of this protein, and then serves as the chromophore. And with these very large degrees of conjugation, you get far red or near infrared emission. And so I want to mention these proteins because they're really a huge advance. They can really improve your imaging. Near infrared and far red fluorescent proteins tend to be quite a bit dimmer than the standard beta barrel proteins. But because they're far red and because red excitation light penetrates tissue much, much better than blue or green, for example, there's far less background. You get better tissue penetration. And it's even possible, since a lot of the emission and excitation wavelengths will end up in the optical window where there's less absorbance by hemoglobin, you can even image some of these proteins through the skin of animals without surgical intervention after injecting virus containing these far red proteins into different organs, and you can use bioimagers to look at them. So those are some of the advantages of far red and near infrared proteins. That's definitely worth mentioning here. So Next, I want to help everybody understand fluorescent protein characteristics, because part of the goal of this talk today is towards the end, I'm going to give very specific fluorescent protein recommendations, but I really want to just show the reasoning and help everybody develop some of the tools you need to interpret data sheets that look like this, make sense of them, and then also think about the other characteristics that are not just captured by studying pure protein. We also want to understand how proteins work biologically and when they're being expressed by a cell, and not just what they look like in a test tube, which can often be quite different. So some of the properties I want to mention, and we can divide these into roughly 3 different groups of properties. I would say when we're looking at proteins in a tube, we're thinking about chemical properties. Then we put them into cells, and we want to understand their biological performance. You know, when we put mCherry into a cell, how is that different than, say, tag RFP? Are there really important considerations here? In many cases, yes. So we'll talk about those. And then the behavior of that protein in its biological environment under stress, for example, when it's put into a 2-dimensional environment that might affect its oligomeric state. Does it really continue to behave like a monomer? Could that lead to erroneous interpretation of results? If it acts like a dimer, how photostable is it? And does it aggregate in the cells, and is it cytotoxic? So these are kind of different domains that are all important to consider. So I want to define 2 terms up front that are reasonably straightforward. One is what the quantum yield, and the next is what the extinction coefficient means. The quantum yield is really just kind of a ratio of the number of photons emitted versus the photons absorbed. As I mentioned in fluorescence, let's say in the case of GFP, shorter wavelength, higher energy blue light goes in, it's absorbed by the protein, and then there's a probability of those photons being emitted in longer wavelength, in this case, green light. It's never going to be more than 100%. It's always a bit less. In the case of GFP, it's about 70%, meaning roughly all things being equal, 70% of the excitation light is going to be emitted as green emission. And then the extinction coefficient, well, like when we're thinking of particles in solution, is really just how much of the light is absorbed versus emitted, and what the capacity of the chromophore is to absorb that light. And then quantum yield is, you know, as I mentioned, what percentage of the photons are re-emitted as light that's useful to your experiments. So that is all about looking at fluorescent proteins on a per molecule basis. So thinking about them in isolation in a tube. So when they're expressed in cells, there can be some big differences. So first I want to talk about monomericity. When it's not always critical that your fluorescent protein be a monomer, but it's usually very helpful. So there are 3 ways that the monomericity of a fluorescent protein are determined. One is with the pure protein, gel filtration chromatography. You run it through a sizing column at different concentrations, and you can determine at what concentration the fluorescent protein mostly elutes as a dimer. Very useful chemically. Honestly, these days, it really doesn't tell you so much about what's going on in the cell and what happens with the protein actually in the environment you're going to use it in. So most people will then try to tag their fluorescent protein under typical experimental conditions to different intracellular targets. Another way that is a pretty decent assay nowadays is called the OSER assay that I'll talk about, that actually puts the fluorescent proteins in a challenging environment that really forces them almost to dimerize, and then you can quantify to what extent they really do. So targeting fluorescent proteins like HyperfolderYFP as an example to different intracellular targets can really show you if there are aggregation effects that occur. You know, and so you can target to nucleus, to tubulin, to actin, clathrin, mito, ER, what have you, and see that the cell morphology looks normal or not. That is a good test. It's very important. Tetramers don't tend to behave well in fusions. Dimers don't usually behave well either. So if a fluorescent protein aggregates in the cell, that can just actually hugely disrupt organelle function. It can restrict the dispersal of the protein, so you won't actually light up the regions of the cell that you really want to see. It can cause mislocalization, and at worst, you might mistake those effects for pathology. So it is very, very important that a fluorescent protein actually behave like something very neutral in the cell. You don't want it to increase cellular processes or facilitate them. You also don't want it to hurt what's going on in the cell. You want it to be a neutral tag. So monomers tend to disrupt cell biology a lot less than dimers and tetramers. I'll show some examples of why. The other thing to consider is that oligomerization, or let's say dimerization, is a concentration-dependent process. So I want to talk about an assay called the OSER assay that is a great demonstration of this. Also, you might note that a lot of fluorescent proteins are prefixed with a lowercase m. That stands for monomer. But many fluorescent proteins were not developed under the same standards. We've had over 30 years now of intensive fluorescent protein development. Standards and ways that we really look at this have sort of changed over time. So some proteins that are prefixed with that M that would make it seem like they're a monomer actually don't behave like monomers in cells. So one assay I want to describe that fluorescent protein developers use is called the organized smooth endoplasmic reticulum assay. It's not perfect, but it's a very good demonstration of what happens, for example, when you anchor a fluorescent protein onto a two-dimensional surface. Normally an FP in space is moving with three-dimensional tumbling motion and has many degrees of freedom. The probability of that interacting with another fluorescent protein molecule then is going to be what it is. But if there's a propensity for that FP to dimerize, that interaction is not really being forced. It may occur depending on the KD of the dimer interface. But what happens is when you express proteins in constrained environments, like on cell membrane, or let's say in the cytoplasmic phase of the ER, anchored into the membrane, the attractive force between those proteins increases a lot because the local concentration of the proteins increases. So this is true for your experiments when you're targeting things to organelles, especially membranes, and it can become very important. So normally the ER has a reticular structure, the FP is well distributed, and we see normal ER morphology. In this assay, again, simply targeting the fluorescent protein to the ER using an anchor motif from cytochrome P450, the fluorescent proteins that are more prone to dimerization will so strongly associate that they will pull on the ER network, disrupting the morphology and leading to these kinds of whorl structures that you can see here from TD tomato, for example, that grossly distort the ER morphology and could make you think you're seeing something pathological. So it turns out that many fluorescent proteins that are weak dimers, even some that are actually monomers, can disrupt organelle morphology when they're targeted to them. EGFP is a weak dimer. Weak dimers can work very well for tagging. You know, there are situations where it becomes a lot more important to use monomers. Targeting things to the ER, to the cell membrane, it really is pretty critical that you use monomers. Even M-cherry can cause some aggregate structures. Citrine, about as dimeric as EGFP, actually causes more problems than EGFP, and that's partly because it folds a little bit differently. Whereas monomeric EGFP shows basically no problems being targeted to a sensitive structure like the ER. So what I want to help with is give you ways to make sense of this, because these are things that aren't so commonly reported in data sheets, but they can have a very big impact on your experiments. And now these kinds of biological-based assays are starting to pick up, and there's a greater availability of this information. So one of those reports is Paula Cranfill's study from 2016, and I'll have some resources towards the end that you can look at, that used this assay to quantify the degree of normal cells after transfection of these constructs. And it turns out that many fluorescent proteins that we use and we treat as monomers are quite monomeric. Monomeric EGFP, mCherry, MRFP1. But EGFP, Clover, and many others are weak dimers. And many of us have used EGFP in fusions, and in most of our fusions, it's fine. So what weak dimers are okay, I'm not saying that it's the wrong thing to use, but you do need to consider the context. So if you're tagging something to the membrane or to other organelles that really constrain the three-dimensional motion of the fluorescent protein, you really want to be thinking about monomers. So many fluorescent proteins that have that lowercase m are actually pretty strong dimers after all. And again, that's because of changing standards in the field. These newer assays can be very effective in showing that. So M cardinal, which is a terrific far red protein that I use all the time, is actually quite dimeric. So I prefer not to use M cardinal, tagging it to membrane, ER, or really mitochondria, for example. M Kate, also a great far red protein, has similar issues. T tomato is a tandem dimer. It's a pseudomonomer, but it still behaves like a dimer in solution. And I would not recommend using TD tomato as a tag in any situation. There are better choices. So basically you can start to get these kinds of aggregation effects. So another thing to consider is the fluorescent the pKa of the chromophore. Almost every spec sheet is going to give you this information. We all know that the cell has many different compartments that have different pH. the really key takeaway here is that if you're going to work in an acidic compartment, as we get more into the secretory pathway, for example, it really behooves you to choose a fluorescent protein that has a low chromophore pKa, let's say below 6. So, I developed a fluorescent protein, a much brighter YFP than EYFP, for example, called HyperfolderYFP. I'll talk a bit more about that later. Its pKa is 5.6, EYFP's is 7, which makes HyperfolderYFP about 20 times less acid sensitive than EYFP. So I wouldn't want to use EYFP in the secretory pathway, but HyperfolderYFP with its low pKa and other stability properties is a much better choice. So if a fluorescent protein has a very high pKa, especially above 6.5 or so, you really want to be thinking whether you're going to see large pH changes in that environment and maybe choose something with a lower pKa. So to talk about maturation and folding, and this kind of gets into, I'm going to talk about some of my research and then other fluorescent proteins that we can learn about through that. Maturation and folding are processes by which, one, maturation is the process by which the chromophore inside the barrel forms, and folding is when the overall structure of the fluorescent protein turns into that 11-stranded barrel shape. So a lot of these considerations come about because fluorescent proteins evolved in coral reef kind of conditions or Pacific coast waters. So, for example, almost all of these are evolved to fold at 20 to 25 degrees, not optimized for 37-degree temperatures like the mouse. So there's been a lot of effort to make fluorescent proteins that have better thermostability and better solubility at 37 degrees. So I've made some contributions to that with mGreenLantern and HyperfolderYFP. HyperfolderYFP is intended to be a direct replacement for superfolder-GFP, and it outperforms it in every domain. And I'll just kind of mention what those differences are, what you'd want to consider, and why you might want to use a protein like this, and why in some cases it doesn't even matter. So something to think about is just when a fluorescent protein is folding, those strands are kind of out in the open. The amino acids on the inside tend to be hydrophobic. They can aggregate. So really, folding needs to happen in a controlled manner, and it needs to happen relatively quickly, but not so quickly that you get misfolded aggregates. And there are some things that can contribute to that. So basically, at this point, the chromophore is starting to mature in the central helix as the barrel is closing. This can even occur co-translationally. Some fluorescent proteins like HyperfolderYFP and mGreenLantern fold so fast that it's likely a co-translational process. So the key thing to understand about misfolded fluorescent proteins is that they're non-fluorescent. So like most proteins, they need to fold properly to work. Well, the way a fluorescent protein works is it takes light of one wavelength and it emits it at another. So for the chromophore to function, the barrel needs to be folded properly, and there are all kinds of kinetic traps that fluorescent proteins can find themselves in to create non-fluorescent, misfolded, potentially aggregation-prone structures. So during my PhD, I had a very simple idea, which is that if two fluorescent proteins have the same molecular brightness, in other words, if that protein in solution is just as bright as another, but the other protein is able to fold more efficiently than, let's say, protein A here, then cells expressing protein B should be brighter, right? All things being equal, if you have the same product, but one product is made more efficiently, well, then cells expressing that should be able to be brighter. So let's say we have something like wild type GFP here, which is actually not terribly soluble at 37 degrees. You might get some properly folded protein out of it, we do, but there's actually a lot of invisible, misfolded, aggregation-prone protein that also results. So it would be ideal to make protein that, even if it were exactly as bright on a per molecule basis, is more efficiently synthesized by the cell. And a lot of fluorescent protein research these days is geared towards simply making better expressing protein by improving its folding. So that's what I did with mGreenLantern. I'll mention that in a moment. And so again, molecular brightness is really the brightness of that protein in solution. Cellular brightness, and what we would call like in-cell brightness or cellular brightness, is the brightness of cells that are expressing that fluorescent protein. And then we compare those to an FP standard. So a way that you might measure this in the lab, and this is what I do in my work, and other labs have adopted this kind of ratiometric strategy, is to express your protein of interest, that's your experimental GFP, in a single plasmid alongside a fluorescent protein control, like let's say mCherry or what have you, As long as the standard stays the same, then you can control other fluorescent, you can compare other fluorescent proteins by transfecting these into different wells. So let's say I want to compare the brightness of EGFP to mGreenLantern or Clover. Then I'm going to, in one vector, I'll have EGFP, and the other, I'll have Clover. All the other components are the same. And then I can take that green to red ratio and compare them to each other. And that's a nice way that controls for transfection effects and other forms of variability and really tightens up experiments. And we quantitatively say, when we transfect cells with this protein, it's really this much brighter than the other. And that's really what it comes down to when we're talking, the results of your experiments. So the properties that contribute to cellular brightness: maturation rate of the chromophore, the maturation efficiency, the copy number of functional fluorophore molecules in that cell is what that leads to. And then you can multiply that by the molecular brightness, and that all comes together to give you what you see in your culture plate. So mGreenLantern is a really good example of this. And I mention it because it's a very bright GFP that I think can really help people's research and has led to a lot of discoveries already. I developed mGreenLantern by improving the folding using these kinds of assays compared to other typical GFPs like superfolderYFP, Clover, mClover3, mNG is mneon green here. These are all very popular GFPs that are widely used, and we all know EYFP as well. So it turns out that mGreenLantern in these assays is up to 6 times brighter than EGFP and maybe twice as bright as mneon green in various experiments. In bacteria, those trends held and were pretty similar. And when you look at this, when I expose these under identical acquisition settings using these kinds of systems, mGreenLantern really outshines the rest. So one would expect, based on the quantum yield and extinction coefficient here, or let's say the product of those, which is molecular brightness, that on a per molecule basis, EGFP should only be about half the brightness of mGreenLantern. So mGreenLantern you would think would be twice as bright. It ends up being something like 3 to 6 times brighter in practice. So there's reason for that, and it does come down to improved folding efficiency, and I'll show some more data on that. In practice, mGreenLantern really enhances neuronal tracing experiments compared to EGFP and EYFP. So it's made more efficiently by the cell. It's brighter. When we inject it into different regions of the brain, we can see expression very, very rapidly, often within just 48 hours, and then about a week to get full expression. About a week shorter than you would get for EGFP to see its maximum expression level. We can see long-range neuronal projections. When we compare under the identical acquisition conditions and in the same vectors to EYFP, injecting EYFP into the brain does not show very long-range projections. Injecting mGreenLantern using the exact same assay, we can see very long, almost 4 millimeter long projections through the corpus callosum, the catecutamen, and other regions. And that was true regardless of the excitation light we used, whether using 488 or 514, mGreenLantern outshined EYFP in every experiment. And it's been very helpful. mGreenLantern also enhances 2-photon imaging, and it's a lot brighter when you use AAV on primary cells. So for example, with EGFP, we always found it necessary to enhance the EGFP signal with antibodies. With mGreenLantern, only 1 week of expression was necessary for us to do good spine analysis. And after 2 weeks, that smoothed out a little further, but was not terribly different than 1 week. So really what's happening is mGreenLantern is it's more efficiently made, so there's just more reaction product that goes into the fluorescent product we want, and there's less non-fluorescent side product. So that better uses the cell's very limited cellular resources, which means you can put it under weaker promoters if you like, because it's so bright. Or you can just maximize that expression as we were doing here and use it to fill fine cellular structures like neurons. Under 2-photon imaging, we were able to visualize more spines with mGreenLantern than with EGFP, because EGFP, under the same acquisition settings, fell below the detection threshold in many cases. So we want to be able to count as many spines as possible for our neuronal tracing experiments. mGreenLantern showed significantly more spines that were visible after antibody enhancement with GFP, but we never needed to do it with mGreenLantern. And just the same in primary cells when we use AAV CAG mGreenLantern compared to EGFP, under identical acquisition settings, mGreenLantern is vastly brighter. So that's what I really recommend for neuronal imaging. There's some other good choices, but this is our current favorite by far. And because of mGreenLantern's very high structural stability, it tolerates some forms of tissue clearing very well. And with collaborators, we were able to inject retro AAV2 into the spine and look at the origins of those descending motor neuron pools in the brain. So mGreenLantern can work well for tissue clearing. We found that EGFP and EYFP don't work well for that whatsoever. So it's a very robust protein that will actually enhance a lot of your downstream applications. I want to mention one other protein before I get a little more general and talk about RFPs too. One is HyperfolderYFP, which is another protein that I engineered, in this case, using mGreenLantern as the template. I developed HyperfolderYFP for extreme thermodynamic stability. It's really hard to destroy this protein. It is the most stable protein I'm currently aware of. And it's very useful for advanced microscopy. So some things you might consider that could impact your experiments positively here is that HyperfolderYFP retains much more fluorescence after fixation with paraformaldehyde than the other GFPs that we tested. So this is important because obviously in histological processing, we almost always use paraformaldehyde 4% PFA first to treat tissue and fix and cross-link all proteins. So what people don't often realize, and that can have a huge bearing on the downstream success of your experiments and the whatever imaging you're doing, is that some fluorescent proteins that are otherwise fairly bright, like M-neon green, are actually quenched very strongly by PFA, losing almost 70% of their brightness. So even if a fluorescent protein starts really bright, if it loses all of its fluorescence from PFA fixation, then you're going to have trouble imaging it later. So HyperYFP retains 75% of its brightness after PFA fixation. Also, mGreenLantern fared quite well, comparable to EGFP and others. And it seems that some proteins like EYFP and M-neon green, which also have the worst thermostability, happen to lose a lot of their fluorescence after PFA fixation. HyperfolderYFP has extraordinary thermostability and can remain fluorescent for over an hour, well, for several hours at near boiling temperatures, which makes it very stable in the cell. So more fluorescent protein can accumulate. It has no cysteine residues, so it can't form interchain disulfide bonds, which is a problem with other FPs. It's basically insensitive to chloride. It has low pKa. You can't denature it in guanidinium hydrochloride, which has made it very useful for protein purification as a sort of affinity tag. And its one disadvantage is that its photostability is about equal to that of Venus, which is widely used. EYFP tends to have extremely high photostability compared to other YFPs. So there are always pros and cons, but if for tissue processing, HyperYFP is very much recommended now. And as you can see, it worked very well for expansion microscopy, showing the same cells at different magnification. We can see very fine actin structures with little loss of fluorescence. Another thing to consider when you're choosing your fluorescent proteins is that not all of them are going to be compatible with anti-GFP antibodies. So just to go over this really quick, I engineered mGreenLantern and HyperfolderYFP from the Euphoria Victoria GFP. So essentially going back to EGFP, clover, and related proteins that are all originated from the Euphoria Victoria jellyfish. So really, antibodies are recognizing epitopes on the surface of the barrel that are common to that lineage, fluorescent proteins of that lineage, right? So with M-neon green, stay gold, and MTFP1, which are other great teal or green fluorescent proteins, those are all from different lineages. So anti-GFP antibodies don't recognize them. And in some cases, there may be no antibodies against these proteins. So I was arguing that mGreenLantern is so bright that you don't need to use antibodies, but they're to enhance the signal, which is true. But there are some situations where antibodies can still be useful. If you're doing immunoprecipitation, if you're doing Western blotting, where you're going to physically denature the protein and lose its fluorescence, it can be very important to use antibodies. So when you're making your choices, depending on what you have available, you might want to think about using an Euphoria Victoria protein like mGreenLantern that is more widely compatible with downstream applications than something like M-neon green or something super new like Stay Gold that has no antibodies for it at all yet. So when we look at the Vector Builder Fluorophore page, which has some great options and also some really older and some kind of off little out there options, it's worth thinking about that. So Hopefully everyone can see my cursor and we can see eGFP here, mGreenLantern, SuperfolderGFP, and Emerald. Those are all Euphoria Victoria derived fluorescent proteins that will work with anti-GFP antibodies. But these ones like COP-GFP, Turbo-GFP, HR-GFP, these are very uncommon GFPs that are not widely used in experiments. ZS green is also another completely different one that actually is tetrameric in cells. Neon green is a terrific GFP with good photostability, but it's also from yet another different lineage. None of those will work with anti-GFP antibodies. So you need to know what organism they came from if you're planning on using antibodies or anything else that might need to associate chemically with the surface of the barrel in a selective manner. So worth considering where they come from, and you can always look those up. So, oh yeah, and then also this is just more data that shows that indeed mGreenLantern and HyperfolderYFP produce much more soluble protein, all other things being equal. When we induce cultures at the same absorbance, we harvest their protein at the same concentration, run them on mossy gel. You can see that HyperfolderYFP and mGreenLantern make by far the most soluble protein in human cells. And then they make proportionally far less insoluble protein. So the theory checks out in practice. This is largely what contributes to the greater brightness as you get more molecules accumulating in the cell, and the cell is a lot happier. So next, I just want to mention RFPs. When we talk about chromophore maturation, there's something that's really important for your experiments here. And I love talking about the chemistry of chromophore maturation and how these all come together, but they're really just like two key takeaways that you need to know that can impact your experiments. The first is that in RFPs, there are more degrees of conjugation needed, which means there are more reaction steps required. And in a chemical synthesis, when there are more reaction steps, it means there are more things that can go wrong. So to make a red chromophore, it actually proceeds through a blue intermediate that then this conjugation process with the benzylidene moiety is what actually happens at the end to make the red chromophore. Green only happens by accident in RFPs. So when some RFPs make a mixture of chromophores, they'll make red and a little bit of green and a little bit of blue. In some cases, for single color imaging, you won't even notice. It's not a problem. But if you're doing very sensitive multicolor imaging, that can present some really big complications. And there are some fluorescent proteins that are much better at this than others. And as I mentioned, fluorescent proteins like mGreenLantern, like the new mScarlet that I'll talk about, and others were really optimized using cellular brightness assays through which the green chromophore could essentially be evolved out and it's no longer present. So in these earlier fluorescent proteins, like tag RFP, mRuby and mRuby2, those were great proteins at the time. In the early 2010s, late zeros, we really didn't have too many other options besides those in mCherry. Nowadays, they really are obsolete. They make too much green chromophore. The tag RFP series in particular, I don't recommend for use anymore because you'll get a mixture of blue, green, and red. And think about it this way. If not only can that lead to bleed through in your experiments, but if you're making more blue and green chromophore when the goal is to make red, it means you're making less red, which means they're going to be less bright than they could be. And so better options, might be mCherry, mRuby3, mOrange, TD Tomato. Those are all great, bearing in mind that TD Tomato is a pseudomonomer. It's more like a dimer. There are always these trade-offs. But really nowadays, the mScarlet series, like mScarlet, mScarlet3, are really just hands down the best. They make almost exclusively red chromophore. When in my experiments, when I test these, I cannot detect any green chromophore, even when I express these very aggressively at 37 degrees. So, FPs have come a long way in 30 years. In some cases, it's good to stick with what you know and what you have, but depending on the kind of experiment you're doing, it might be time to upgrade. mScarlet came from cellular brightness screens, as I mentioned. It's a lot brighter than mCherry. It's faster maturing, and really it surpasses mRFP1, tag RFP, Fusion Red, and it really does directly replace mCherry now. mCherry is still a terrific choice. If you keep using it, if you have a ton of constructs, you're not having any issues, don't need to upgrade, it's fine. But if you're thinking about making new constructs, you want to maximize brightness, perhaps you want to dial down promoter strength, you could use mScarlet and enjoy all of those advantages and never have to worry about it. Um, let's see, I think I was going to talk about some more unusual fluorescent proteins, but maybe I should save this. I think I'll just breeze through it real quick. I just want to mention that there are some fluorescent proteins that have a large Stokes shift. Um, Normally, when we're thinking of three-color imaging, we're exciting with three different lasers. Let's say a blue protein like mTag-BFP2, a green like mGreenLantern, and my favorite RFP, mScarlet I3. Use three different lasers. We collect the emission with bandpass filters. A fluorescent protein with a large Stokes shift can be very, very useful, and it comes with pros and cons. The Stokes shift is just the difference between the excitation and the emission peak. So I developed a fluorescent protein called LSS-mGFP, large Stokes shift monomeric GFP, because there was a need to be able to open up another color channel. So red, green, and blue sometimes isn't enough. You can use far red, but sometimes you don't have far red on your scope. Maybe you have a highly configurable scope and you can change your filters. So in this case, LSS mGFP is exclusively excited by the 405 line, and it emits green, as opposed to most 405 excitable FPs that emit blue. So that means you can use it with mGreenLantern, which is not 405 excitable, and essentially have two green colors in your set and detect it with high brightness. The nice thing about LSS FPs is because the excitation peak is so far from the emission peak, you can collect the entire emission curve, which means more brightness. Whereas with other fluorescent, almost all other fluorescent proteins, the excitation and emission peaks are very close together, and you have to band pass out more than half of the emission peak, which means you're collecting fewer photons by comparison. So really great. You can open up a whole nother channel this way. There were existing, Turbo Sapphire is another LSS GFP people have heard of, but the problem is it actually can be excited by 488, and also EGFP can actually be excited by 405 and will emit green. And when you excite, if you target EGFP to actin and Turbo Sapphire to the nucleus, you will excite Turbo Sapphire, and you can actually see it right here where it shouldn't be. It's being excited in the nucleus, and you can actually see that through overlay. I developed LSS mGFP to completely eliminate that problem, and you can use these together now for artifact-free imaging. And you see that halo totally goes away with LSS mGFP because it can't be excited by 470. And similarly, mGreenLantern cannot be excited by 405. So that's the difference. You can get much cleaner imaging, less cross excitation if you understand what these curves mean and look like. And you pick some newer options like the ones I'm recommending here, as opposed to these that are over 30 years old. There are other LSS FPs, and towards wrapping this up, I just want to give some more just discrete tips. One is it's very tempting to use a lot of colors for multicolor imaging. We have better vector systems today, thanks to, and it's easier than ever to do, thanks to companies like Vector Builder. I would always recommend using the brightest fluorophore to tag your lowest abundance protein or your most challenging target. So just some simple reasoning here. If you have less of your target protein in the cell, or it's less efficiently synthesized, you want to use your brightest protein to tag it, so that on a per molecule basis, you're going to have more photons hitting your detector, and you're going to be able to see that protein better. So mTag-BFP2, for example, is a BFP. It's a pretty good one. But as a BFP, they tend to be kind of dim. So you want to tag your highest abundance protein with that. And then you should actually get, hopefully you should get a nice ability to resolve both targets. If we put our brightest protein on the highest abundance protein, it's going to be way brighter proportionally than putting your dimmer protein on the lowest abundance one. So just some simple reasoning. Dimers are fine for cytosol, but otherwise you really should use monomers. The reason people use dimers a lot in the past is simply because dimers tend to be brighter. When the fluorescent protein is stuck alongside another one, it traps the light better, so to speak. It has a larger optical cross section, and usually that improves things. It makes it brighter. But now monomers have been engineered to be so bright, it's really worth just sticking exclusively with monomers. There's really no downside. Dimers are not reliable fusion tags, so don't tag stuff with TD tomato. Great for the cytosol, not great for fusions. And only tag what you really need to see. It's harder to transduce and transfect multiple payloads at once, and every additional overexpressed protein consumes cellular resources. So choose carefully. If you want to conserve cellular resources as much as possible, use the brightest protein, and you can dial down the expression level using a weaker promoter so that you're making less protein, but it's going to be brighter, and that will stress the cell less. There are two, so kind of as we get towards the end of this, I want to mention two new fluorescent proteins. I've mentioned how mGreenLantern is a great GFP, mScarlet is a great RFP. Well, there are some new proteins that have some other advantages in involving extreme photostability. These are not currently on Vector Builder. They're quite new. That's why they've only been made in the last couple of years. One is called Stay Gold. Stay Gold came out a couple of years ago. It is by far the most photostable fluorescent protein out there. It was actually mind-blowing. I reviewed one of the papers for this, and it took quite some time to convince me that this was really true. I had to try it myself, and it really does work. So Stay Gold is amazing for intensive continuous live imaging. If you're looking at fine structures like neurons or mitochondria, and you need to image them for hours at a time, you can't do better than Stay Gold. It's surprisingly bright for its photostability, which usually comes with a brightness trade-off. So Stay Gold is a dimer. So you want to be careful where you use it. There are some new monomeric Stay Golds coming out, but you should test these carefully because they are new FPs and you want to make sure that they don't disrupt your assay. So always do a comparison. Take your favorite FP and then drop in, say, Stay Gold next to it and make sure it performs properly. It should do quite well, but you always want to know in your particular assay. mScarlet3H is a new mScarlet3 point mutant that makes it the most photostable RFP, but it is 5 times dimmer than mScarlet and mScarlet3. So if you are going to just be using routine imaging and you just want to see your target, definitely prefer mScarlet and mScarlet3. If you need to do time resolved imaging or continuous videography, then yeah, you might want to consider one of these ones. But otherwise, you know, stick with the basics. They work really well. I won't dive into photostability. I just want to mention if you're doing really intensive long-term imaging, the most important factors in photo bleaching are time and intensity, and they can vary so much between what you see in different papers. Sometimes you just got to compare two fluorophores side by side. That's really the take-home message. So I mentioned various things here. When you're choosing a fluorescent protein, and you really need to think, why am I going to choose this compared to others? Say you're setting up a new vector system, you want to take a fresh look at what you're working with. You really want to just look at the most rigorous head-to-head comparisons that are going to be the most generalizable across papers. A lot of papers will just compare their new FP to like eGFP or another one. But you want to look at some of these more comprehensive studies where many FPs are compared side by side in the same experiment using the same methods. And so these are some of the papers that I would really recommend reading. You know, my GFP and YFP paper is here where I rigorously compared all of the best GFPs and YFPs at the time against each other. Daphne Bindle's paper on mScarlet, the mScarlet paper, is a great comparison of RFPs worth looking at. There's a good near-infrared FP paper where they compared at least 12 different near-infrared FPs together. And then Paula Cranfill's 2016 Nature Methods paper compared all of the best FPs up to relevant to 2016 under the same assays. So that can be very worth using, especially for OSER data. Okay, last couple minutes here. I'll mention, you know, looking at the vector builder page, there are some really good choices here. I've mentioned how some of these more unusual GFPs come with pros and cons. They're not very widely used. mGreenLantern, EGF, or the ones that are much more widely used would be mGreenLantern, eGFP, Neon Green, and sometimes HyperfolderYFP. These other ones are really special case GFPs, and you may have reasons for using them, but just again, they're not compatible with standard GFP antibodies, that you'd have to use selective ones if they're even available for those. There are some slightly outdated FPs here, like YPET and SciPET, which were originally designed for FRET. So these are quite dimeric. If you want, yeah, so these are the ones that I would actually, in yellow, that I would recommend probably avoiding and choosing newer options. For YPET, you could use mVenus, which is a terrific choice. That's already on the list, or HyperfolderYFP. For SciPET, mTurquoise2 is, if you need a cyan FP, use mTurquoise2. It really is phenomenal. We haven't been able to do better than that. But again, dimers can be totally fine. These are on the list likely because they're really bright, which is true, and that's partly because they're dimeric. But again, thinking about the pros and cons of a dimeric FP, I wouldn't use these to tag your protein of interest, is what I'm saying. If I needed to tag an endogenous protein and I wanted cyan, I would use mTurquoise2. So that's some of the difference. mScarlet3, wonderful RFP choice, probably the best out there. MIRFP 670 Nano and Nano 3 are my favorite near infrared fluorescent proteins. And, MApple and MK2 are still solid choices. So, Vector Builder has a good list here. There's nothing wrong with any of these. You could do quite well. It's just important to understand the pros and cons and how that's going to be relevant for your experiments. So wrapping this up, I have made my own best-in-class fluorescent protein recommendations as of 2025. I'm going to just spend maybe 2 minutes going through it, and then I'll sum up because we're getting towards the end here. As far as BFPs, MTAG BFP2 is the brightest. Just note that it's not actually a monomer. So again, if you're tagging a protein, it doesn't mean you can't tag a protein with it, but if you're looking at something very, very delicate like the ER or cell membrane, I really would prefer eBFP2, and especially the monomeric version. It's really the only monomeric BFP so far. But again, BFPs tend to be dimmer than the rest. When we're looking at the extremes of the visible spectrum, that's where the quantum yield tends to be lower. So if you need cyan, mTurquoise 2 has been the winner for over a decade now. If you needed something in between, MTFP1 is quite good. I mentioned Stay Gold, which is very, very bright. It is a tandem dimer, so you have to consider that, kind of like TD Tomato, and it's incredibly photostable. I mentioned LSS mGFP if you need a 405 excitable GFP. mGreenLantern is, obviously, I designed that, so I'm a big fan. It's extremely bright. It expresses very rapidly. It's compatible with EGFP antibodies. It's good for some forms of tissue clearing with expansion microscopy. An all-around terrific choice. The only downside is that I wouldn't do intensive live imaging with it. Let's say you're trying to record mitochondria for an hour at a time. Really not the best choice. I would prefer Stay Gold because of its high photostability. But for routine imaging, because mGreenLantern is so bright, you don't even have to use very strong excitation light, which means you're going to get lower background. So as an all-round FP, mGreenLantern is my choice. I'd also recommend mNeon Green. mNeon Green is still an excellent option. For yellow, monomeric HyperfolderYFP is my personal favorite. If you want to try something a little bit more traditional, mVenus is also excellent. And you can find all these stats on fpbase.com or from the original publications. For orange, mOrange and mOrange 2 are terrific. And then really any of the mScarlet proteins are going to be fine. If it has an H at the end, that means it's the more photostable version that's going to be dimmer. If it has the little as the I in it, that means that it's a faster maturing version. I like the faster maturing RFPs. So mScarlet I and mScarlet I3 are my favorites. But mScarlet, mScarlet 3, all these versions, you really just can't go wrong. And it's what I would recommend by far the most for RFPs. For far red, if you need a straight far red protein, mCardinal is my favorite. It is a bit of a dimer. For near infrared and super far red, MIRFP 670 Nano 3, which I know is a mouthful, is a really amazing protein, and it's only about 140 amino acids, making it smaller than all the rest. Great for viral vectors. MIRFP 713 is kind of the best near infrared in my opinion. High brightness, you can even detect it on a LiCor scanner, which is really fun to do. So these are kind of my recommendations. Doesn't mean that other proteins won't work for you, but I would say if you're designing new vectors and you're trying to assess your experiments and you have these options, I would go with one of these in most cases, and they should be the most versatile for you as well. So just to summarize, EGFP came from the Aquaria Victoria jellyfish, like Clover, mGreenLantern, HyperYFP, Venus, many proteins. The Chromophore is sort of like a candle and a lantern. Optimizing fluorescent proteins for better folding and maturation makes them brighter in cells and makes a big difference and helps improve cellular resources quite a bit, meaning the cell is going to experience less cytotoxicity, it's going to divide properly, and you're going to get better signal. So it's especially if you're trying to do things involving gene therapy, you're trying to look at toxicity while using a reporter, you really want to use these optimized, better folding proteins. They're going to cause you way fewer problems. And FPs have come a long way in the last 30 years. There are lots of choices now. I hope I've given you some of the tools and information you need to make the best choices. Again, reinforcing that a lot of the traditional FPs are still great. EGFP is fine. You know, there is something to be said about a tried and true tool. You don't just want to rush to upgrade to the next shiny thing, but I hope that I've explained why some of these newer FPs are worth your time. And also just pointing out the pros and cons of dimers and monomers. And just now in 2025, the highly engineered monomers are really just the better choice in most cases, except for the weird ones like Stay Gold that offer the massive photostability advantage. And also just a shout out to FPBase. FPBase is a really good website. You can learn a lot about fluorescent proteins from there. There wasn't time to kind of go through that, but everyone can, you know, you can look at fpbase.org on your own, and it's really terrific. I like their spec reviewer. So thank you so much. Really appreciate you guys taking the time. Big thanks to Vector Builder for bringing me on today and to everyone in attendance. You can get in touch by email. I'm happy to help with fluorescent protein questions. I'm on social media here and there, mostly to share new work involving FPs. So feel free to keep in touch. And again, thank you to Vector Builder. I really appreciate it.
Thank you so much. That was truly incredible. Wonderful information about so many different fluorescent proteins. We have a couple of questions, and we may only have time for a couple. Here's your first one. For in vivo applications, what about the immunogenicity of different fluorescent proteins? Is mCherry less immunogenic than EGFP? That's a common, frequent comment I've heard.
Yeah, that's a terrific question. When I was in the gene therapy field, that's something we wondered about a lot. There have been some efforts to decrease the immunogenicity of FPs. So some people are studying that, but I honestly would not say there's enough really good data on that. If you're worried about that, then yeah, sticking to some of these more traditional FPs might be the best choice. But it's not something that developers really study. That's a more specialized question and extremely relevant to gene therapy. And honestly, I would like to see more experiments done on it. I would like to speculate, but I'd rather give you a concrete answer. And I don't think I can give you a solid answer on that here. So yeah, even EGFP has immunogenicity hotspots that can be mutated out, but we really need to see some better studies on it to say concretely.
Thank you. Have you assessed which linkers work best with some of these new fluorescent proteins, same as EGFP and mCherry, or something different?
Yeah, so in most cases, the N and C termini of the fluorescent protein are the same. So a lot of fluorescent proteins developers, like such as myself, like almost all of them actually use the same seven N-terminal amino acids and same seven C-terminal amino acids as on EGFP, which means, you know, and with the barrel structure being very, very similar, the same linkers tend to work really well. It only changes a little bit when you're working with something really new that has radically different termini, like Stay Gold. And then I would just go with whatever's in that paper. If you go to fpbase.org, you can see what the amino acid sequence looks like. And if the termini look the same as EGFP, which it will in most cases, you can be pretty confident about your linker choice, just being whatever worked for that among the most common plasmids. M-scarlet proteins have slightly different termini, but those work really well with traditional linkers too. So in most cases, the same linkers will do just fine.
Awesome, thank you. Crimson R is normally tagged with D tomato, not TD tomato. What are your thoughts on this?
Yeah, so that's a straight up dimer. So depends what you're looking at. You know, it is going to be dimeric. You know, if I were looking at something like actin, you know, or other fine filaments, I really wouldn't want any attractive force between the fluorescent proteins to be, you know, significant in any way, because that can actually, like I showed with the ER data, that can really pull those fluorescent protein units together, and then the symmetry related, you know, dimer is going to form, depending on that context. So I think the D tomato probably is just because it's a dimer, which makes it brighter. That might have been the best technology at the time. You would do even better to use M-scarlet, for example. It doesn't necessarily mean that experiment is going to be wrong or produce errors, but definitely just consider the overall context of what kind of structure is this? Am I decreasing the ability of this fluorescent protein to tumble in space and restricting it to a two-dimensional surface? If so, then the propensity for aggregation is going to be greater. And if you see speckles and other puncta, that might be something, might be worth considering replacing your FP.
Our next question is, in your opinion, why is the tandem dimer aspect of stay gold not a liability compared to the other dimeric FPs?
Yeah, I mean, so it is a liability in some cases, just the same as TD tomato or D tomato. I think we give stay gold a little bit of wiggle room because it's so photostable, but the other ones, there is an M stay gold. So there is a truly monomeric stay gold that was developed I think from Miyawaki's group, and then Kirill Pyakovich and Fedor Subak's group, among others, developed M-Baojin, M-B-A-O-J-I-N. That is a monomeric version. So if you're concerned about that and you still want the pretty high photostability and brightness, you could try one of those. You can find those, on fpbase.org too, by looking up stay golds and then looking at the kind of lineage tree. So you do have options for that, and it is worth considering that any pseudo monomer, which is a nice way of saying, tandem dimer, may have problems. It's bulkier, which means it has a larger effective radius. Even if the dimer interface is tethered together and designed to kind of self-associate, there are still residual interfaces that can associate, and then you have sort of a tetrameric thing going on. So yeah, you do want to be careful with it. I should make that clear.
Thank you. So in the interest of time, maybe we'll do one last question. I'm sorry if your question wasn't answered. I'm sure you can reach out to the speaker if you have more questions, or us at Vector Builder. Here's the last question, I guess. Is there a mGreenLantern based G-Camp or other sensors D-Light in the works?
Oh, that's a good question. So I actually made a variety of mGreenLantern mutants that you can find in that paper and in the HyperfolderYFP paper. And then in the HyperfolderYFP paper, I solved 3 high-resolution crystal structures of an mGreenLantern variant and HyperYFP. And people are starting to use those because they're good crystal structures, and these are well-folded proteins of high stability, high brightness. It does actually make them optimal templates for sensor engineering. So the mGreenLantern variant called Fold 6 is one of them that we have a 1.2 angstrom structure of. HyperYFP itself is being used. And so actually I'm in Lauren Luger's lab right now, where a lot of these calcium sensors originated, and we are using those to develop sensors. So they actually, because the sequences, the residues inside the chromophore region are different, they actually give you some more variety in developing sensors. So I would say, some of this is in the works. I think some people are making split fluorescent proteins with them too. But since they're relatively new, it takes time to build sensors, but there definitely is activity and interest in that. So you can expect to see probably more of those, and you can also use them yourself to develop sensors. Yeah.
Wonderful. All right. Thank you so much for speaking today. This was truly incredible. And thanks everybody for attending today's talk. I hope you learned a lot. And hopefully we'll see you next month for our next Gene Delivery Seminar Series. Bye, everybody.
Thank you so much.